Hello and welcome to part 5 of our Origin of the Eukaryotes lecture. So we've talked about how all of these eukaryotic supergroups have mitochondria, and the last universal common ancestor of the eukaryotes had mitochondria. And now let's talk about the chloroplasts. So we know that plants have chloroplasts, and it turns out that all of the plants that you know about, all of the flowers, all of the crops, all of the cacti, all of the trees, including conifers and angiosperms, all of the plants are in this one branch in this phylogeny that also includes all of the green algae. Because taxonomically, land plants are basically just very large green algae. They have the same recent common ancestor. A cousin to the green algae and the land plants are the red algae, and there are also these weird algae called glaucocystophytes that together all make up the supergroup called the archaeplastids. Archaeplastids is a weird word that means old plastids, and plastids is a general term that includes chloroplasts and other organelles like chloroplasts. Okay, so it means old chloroplasts to get at the idea that the common ancestor of all of the groups in this group inherited a chloroplast from an ancient ancestor. Okay? But these are not the only organisms that have chloroplasts. All of the taxa on this tree in green font have chloroplasts. And we call eukaryotes with chloroplasts algae. Now, at one time, biologists thought that the algae were a coherent group. It makes sense, right? They're eukaryotic. They can do photosynthesis with their chloroplasts. There are many similarities, many of them being that they're kind of green, and many of them are single-celled, but not all of them. And now we know that it's not true. The algae are not monophyletic, and today the word algae actually has very little meaning, except what I have on the slide, that they are photosynthetic. And the evolution of all of these different algae groups is actually a really interesting story, and it continues our theme of symbiosis. So let's talk about that. How did all of these different lineages in the eukarya get chloroplasts? So there are a couple of different potential explanations, and let's think about them. One potential explanation is that the common ancestor of all of these groups, which would not necessarily need to be all of the eukarya, because there are no algae in these two groups, but it would have to be the common ancestor way back here, which is still pretty far back. If that ancestor had chloroplasts, and then all of the descendants from that ancestor had chloroplasts, and then some of these taxa that do not have chloroplasts today subsequently lost their chloroplasts. That is one potential explanation for the distribution of the chloroplasts we see, where some of the groups, some of the taxa in each of these superkingdoms have chloroplasts and some do not. Okay, so that's one possibility. Ancestor had chloroplasts and then it was lost multiple times independently in multiple superkingdoms. Okay? A prediction of this hypothesis would be that if you made a phylogeny of the chloroplast genes, because remember the chloroplasts have their own genes, and right now we're looking at the genes in the normal genome of these organisms. But if you looked at the phylogeny of genes that you got specifically from their chloroplasts, then that tree should look pretty much like this tree of the eukaryotic genomes. Okay? Because it's the same phylogeny. It's the same ancestor. Everything's happening the same way. That prediction might make more sense if you think about a different potential explanation. This is our potential explanation number 2, which is that the common ancestor of these algae did not have chloroplasts. And the reason why all of these taxa have chloroplasts today is because they independently acquired them. For example, these diatoms and brown algae got their chloroplasts from a different cyanobacteria than the cyanobacteria that gave rise to the chloroplasts in the green and red algae. And then the dinoflagellates and apicomplexans got their chloroplasts from a different cyanobacteria. The cercozoans got their chloroplasts from a different cyanobacteria, and so on. Okay, so that's clearly one potential explanation. If the red algae and green algae could enslave a cyanobacteria and convert it into a chloroplast, then why couldn't these groups do it too? Right, so that is one potential explanation. And the prediction of this hypothesis would be that if you made a phylogeny of the chloroplast genes, you would show different cyanobacteria popping up in the phylogeny of these algae as close relatives of their chloroplasts. So instead of just one cyanobacterial ancestor, you'd have multiple recent cyanobacterial ancestors showing up on the tips of the tree. So that would be the prediction that you could potentially test. Potential explanation number 3 is that the chloroplasts in the red and green algae were transferred by some horizontal mechanism to these other groups of algae. So it was not in the common ancestor of all of them, and it was not independently acquired. It was acquired once and then subsequently transferred to each of these groups. And the prediction here would be that a phylogeny of the chloroplast genes would show a mess, a mixed bag, where the chloroplast genes would be mixed together in a pattern very different than the tree that you see here for the superkingdoms. That is, that the phylogeny of the chloroplast genes in these algae would not at all resemble the tree that we're seeing here for the eukaryotes themselves, for their genomes. So let's test these ideas. So here's the answer. This is the phylogeny of the chloroplast genes, or at least one version of it. This is the real result of researchers' efforts to figure this mystery out. So let's walk through it. So first of all, we see only one part of the tree that has cyanobacteria, and this deeply branching ancestral lineage here. So the cyanobacteria are clearly showing up as an ancestral branch and are not showing up multiple times in the phylogeny. Exactly, one ancestor of cyanobacteria. One cyanobacterial ancestor. Okay, next thing we can notice is that there's a big split between the green algae, including the land plants, yeah, and the red algae. So the red algae are up here, and the green algae are down here. And the next thing that we can notice is that these other groups of algae are split into those two green and red groups. So the euglenids, which are part of the superkingdom Excavates, and the chlorarachniophytes, which are members of the rhizarians, are mixed in with this group of green algae chloroplasts. And the brown algae and the diatoms and the dinoflagellates and the cryptophytes are all grouping closely in this red algal chloroplast lineage. Now, does that tree look at all like our tree of the superkingdoms? No, right? All of these other algae are now mixed in with the red and green algae, which in this tree form one small group all by themselves. And now here, these other algae are splitting themselves among a green group and a red group. So clearly at least one and maybe two of our potential explanations can be disproven, right? It's definitely not the case that this tree looks just like the superkingdom tree, which would be potential explanation number 1. You can't come up with this tree by these lineages just losing their chloroplast genes. It would not look like this. It also doesn't really look to me like potential explanation number 2 works, because then you'd have to have multiple cyanobacterial lineages mixed in throughout here if you had multiple independent acquisitions. So that really only leaves explanation number 3, which is transfers of the chloroplasts from the green algae to other algae in the green group, and transfers of chloroplasts from the red algae into other members of the red group. Yeah, pretty cool, huh? And that is our current understanding. Explanation number 3. So this is how we think it would have worked. So here is the original eukaryote that came along and engulfed a cyanobacterium that became a chloroplast. We call this the primary endosymbiosis, meaning it was the first endosymbiosis, because there are going to be more. Okay, so here we have a photosynthetic eukaryote. It's got its nucleus, it has mitochondria, it has a chloroplast. Now, here comes along another heterotrophic eukaryote. In this particular diagram, it belongs to superkingdom Rhizaria, so it's called a rhizarian eukaryote. That's one example. There could be others. It comes along and engulfs this whole green alga. In this example, this photosynthetic eukaryote is a green alga. This green algal cell is now fully inside this other eukaryotic host. When this process is completed, we now have a secondary endosymbiosis, where we have a eukaryote that has its own genome, like any other cell. It has mitochondria, like any other eukaryote. And it has within it another whole other eukaryotic cell that has its own nucleus and its chloroplast. And we can actually observe this with many of these algae, these photosynthetic eukaryotes. You can look at them under the microscope and you can actually see the vestigial nucleus of the eukaryote that was engulfed by the other eukaryote. And you can sequence the genome of this vestigial nucleus and directly prove by sequencing its genes where it came from. So in many of these algae, you not only have bacterial DNA in the chloroplast and bacterial DNA in the mitochondria, you have eukaryotic DNA in the nucleomorph, that's the technical term for this vestigial nucleus of the eukaryote that was engulfed, and you have the DNA of the host cell itself. And so Lynn Margulis's radical idea of symbiosis as the central feature of the eukaryotic cell is even more radical than she could have imagined, because not only can you explain the presence of the mitochondria and the chloroplasts as a symbiosis, many of these eukaryotes even have endosymbionts of other eukaryotes inside them. And so you can think of many of these eukaryotes as Russian dolls, nested Russian dolls with multiple layers of membranes and nuclei nested within each other. Pretty wild, right? So let's take a minute to think about this and review. So how did all of these superkingdoms get chloroplasts? Did they all descend from parent to offspring from a single common ancestor? Were there multiple independent acquisitions of cyanobacteria in each of these lineages that became separate chloroplasts? Or was there a transfer of whole cells from one superkingdom into the other superkingdoms? Pause the video, think about it. Which is the correct answer? The correct answer is C. Even though if I had asked you this question before I had explained to you how it works, I doubt that you would have chosen C. I certainly would not have expected C, transferring whole cells. Previously in this course, we've talked about horizontal gene transfer, transferring genes between cells. That's already pretty crazy. This is a horizontal cell transfer. Whole cells are being engulfed and therefore transferred between superkingdoms in this process, which is crazy. There's no way I would have ever expected that to be the case if I hadn't done a lot of reading and learning and studying about it to learn that that just, in fact, is the case. Biology is wild. Okay. And furthermore, I showed you the phylogeny earlier, and it turns out there's really good evidence now that all of these algae acquired their chloroplasts by engulfing either red algae or green algae. And so you can classify all of the photosynthetic eukaryotes as either being part of the red lineage or the green lineage, which was shown in the phylogeny that we saw earlier, and there's even much more evidence than was apparent in that one tree from the book. Okay, that is part 5. I'll see you in part 6.